we're going to start here with Brooklynn Hamilton, who's come to give us the real-life perspective about what it's like to have type 1 diabetes.
Hi, everyone. So my name is Brooklynn Hamilton, and I just want to thank you all for having me here today. I'm 13 years old, and 10 years ago, I was diagnosed with type 1 diabetes at the age of 3. I know there's a lot of scientists in the room, but I know there's a lot of confusion between what type 1 diabetes is and what type 2 diabetes is. So basically, type 1 diabetes is an autoimmune disease. My immune system misfired and attacked my pancreas and killed off the beta cells. I'm no longer able to produce insulin. I did nothing wrong, and no, I did not eat too much sugar. I just like to put that out there because I get that question so frequently. My pancreas no longer produces insulin, and without insulin, I would die. That's why I wear my insulin pump, which I've gotten very good at hiding under my dresses and my skirts over the past few years. I also check my blood sugar 8 to 10 times a day. Over the course of the past 10 years, I've checked my blood sugar over 35,000 times. Through trial and error, my parents and I try hard to keep my blood sugar between 80 and 150. And let me tell you, that's really hard. It's like 1 minute you think you have everything under control. The next week, my blood sugars are everywhere, and we have to start all over again. The problem is if I give myself too much insulin, that's when bad things start to happen. I could go into a seizure, and it could potentially lead to death. As you can see, insulin is no cure. This is why companies like ViaCyte are so important to my family and I. If their technology works, I could have a simple surgery and have a device implanted under my skin. I'd be worry-free of checking my blood sugar, going high, going low, and putting on pump sites. Instead, I'd have a 2-hour surgery every 18 months. Please, sign me up for that, because I would much rather do that. My dad and I are both huge sports fans, and we both share the same love for quotes. And one of my dad's favorite sports figures is Michael Jordan, so I thought I would share a quote with all of you on success. I have lost over 9,000 games in my career. 26 times I've taken the winning shot and missed. I have failed more than I have succeeded, but it's because of those failures I ultimately became a success. Michael Jordan knew something about failure and success. You have to try, and sometimes you will fail. But it's because of all of you are trying and you keep taking that shot, someday we will succeed, and people like me will be cured. Thank you.
Thank you, Brooklynn. So I'm going to give an overview about the encapsulated cell replacement therapy that we're developing at ViaCyte. Treatment of type 1 diabetes through a cell replacement strategy it really has good proof of principle that this will work from the field of islet cell transplantation. So here they isolate islet cells from a cadaver pancreas, infuse them into the liver, and as this has developed over the years, current state of the art is many patients could live 5 years without any insulin in a therapy like this. But the problem is they have to trade insulin needles for lifelong immune suppression. So that means only the most severe of type 1 diabetes patients are eligible for a therapy like this. Not only that, the shortage of donor pancreases, the cost of this procedure, really make it not a viable therapy for the million type 1 diabetes patients or more that are present in the US today. So our approach aims to address these limitations of islet cell implantation. And the product we're developing is a combination product. We call it VCO1. And effectively, it is a cell therapy, but we're delivering those cells inside a macroencapsulation device. the core elements of this approach to address those unmet needs of islet cell transplantation are first and foremost a renewability of the cells that we're going to use for therapy. So that renewability comes from human embryonic stem cells. We have a single embryonic stem cell line that was derived from a single donated embryo that literally has the capacity to make hundreds of millions of patient doses over time. So this is the unmatched power of the proliferative potential of human embryonic stem cells. Those stem cells, of course, are not the therapy, so we need to harness the differentiation potential of human embryonic stem cells, and we worked over a number of years to develop a procedure whereby we can take human embryonic stem cells and turn them into a pancreatic cell population. These cells are not yet islets, they're not yet the beta cells that Brooklynn needs, but after delivery to the host, currently the animals and ultimately, hopefully, the patients, they go on to become islets. And that delivery, as I mentioned, is inside an encapsulation device that we call Encaptra. And the principal role of this device is to protect the cells we're delivering from the immune destruction that would occur by the patient's immune system, either because it's not matched to them, these are allogenic cells that we're using, it's an off-the-shelf product, or in the case of type 1 diabetes, there's also autoimmunity that we need to protect these cells from. So as part of the CERM diabetes disease team, we've been working with Jeff Bluestone's group at UCSF and his colleagues to characterize these immune protective properties of the encapsulation device. And this is a mouse model, so we're putting mouse cells into mouse here. So this is a diabetic mouse. We implant a mouse cell line inside this device, give it to the diabetic mice, and what you can see is over the course of really just a couple of weeks, we can correct the blood sugar in these mice. So these are mice that are implanted with cells that are not from that mouse, so they would be rejected in an allogenic fashion. And this is a diabetic mouse that also has autoimmunity on board. So you can see from this study that the device is capable of protecting an allogenic cell source also in the face of concurrent autoimmunity, which is really the hallmark of what it needs to do to be effective for type 1 diabetes therapy. The other aspect of this encapsulation device is that it needs to be biocompatible, biostable, and because of the cells we put in it, it actually exhibits an astounding level of vascularization. And this is very important. It not only keeps the cells alive inside, but it's really important to very rapidly have the cells inside the graft sense blood glucose as blood glucose rises, and then release the insulin and have that insulin get out from the graft and circulate throughout the body to subsequently lower that blood glucose. And this is really the synergy of the cells that we're putting inside. Because they're derived from human embryonic stem cells, the pancreatic cells are kind of fetal in nature. And as fetuses are growing, one of the main things that you're doing as you're growing organs is growing additional blood vessels to support that organ growth. So that's effectively what is happening in this product as we place it under the skin. In cross-section, if we look at this on the left, what you could see is that the functional unit of the device, if you will, is this membrane that excludes the host immune cells. It also retains our grafted cells inside. And you can see how these blood vessels crawl right up against the membrane. They're there to feed the cells. The cells inside are calling for them to be there, saying, we need these nutrients, please come close. So that's how the graft looks. And then on the right-hand side, we have immunofluorescence staining, just showing that we're not only making the beta cells, we're also making alpha cells, delta cells. All the endocrine cells of the pancreas are made inside this device, despite no direct vascularization, no contact with the host. So it really does become like a small ectopic organ, endocrine pancreas organ. So in mice, we're able to model this. Effectively, what we've done here is a study where we administer a chemical called streptozotocin, labeled STZ. This kills off the beta cells in the mouse pancreas. About a month later, after we establish that these mice have no endogenous regeneration, we can then apply the VCO1 product. It takes about 2 months for the cells to finish their differentiation and maturation, whereby they start to control the blood glucose in these mice. It's also important to note when we implant into normal mice, those mice have blood sugars of about 150 mg per deciliter. So that's hyperglycemic from a human standpoint. So in both of these cohorts of mice, we see that the implanted graft lowers the either hyperglycemic condition or even the mouse blood glucose more towards a human set point. This is just evidence that the beta cells inside this are faithfully human, they're functioning normally, and they're able to control blood sugar in a very normal human way. After 5 months of stable blood glucose correction and control, we can explant these grafts from the mice, and we see that their blood glucose reverts to the expected position, either normal mouse blood glucose if they have endogenous beta cells, or frank hyperglycemia if they were previously depleted with STZ treatment. So we've been working on this product for over 10 years, and we're now nearing the phase of getting into the clinic. So our planned clinical implementation is that this will be a subcutaneous implant. We will likely start by implanting on the lower back or the flank, because as Brooklynn could attest, most patients would be injecting insulin in their abdomen or their thighs, and we really can't afford the risk that they would pierce this device with a needle. It would cause the rejection of the cells inside. So we're going to stay away from those areas of the body. But it's going to be a simple subcutaneous implantation, likely to be done in an outpatient procedure. The fact that it's just under the skin allows us to image this device using very non-invasive technologies like ultrasound, and that's what's shown here in the lower right-hand side. Because we're starting with pluripotent stem cells, the regulators are very concerned about the potential to form a benign tumor that's called a teratoma. If that were to occur, although we do not see this happen in our animal studies, we would be able to detect it with this ultrasound. We can see the lumen of the device expanding, and then we could explant this product. And that's a real big safety advantage on the whole. So most people, when you're talking about cell or gene therapy, if you deliver a therapy, all bets are off, right? It's more or less a permanent condition. So you have to live with any side effects that occur in the hope of getting that therapeutic benefit. But in this unique case, we can explant a graft should the need arise due to toxicity or tumorigenicity. So the path to the clinic, we've had a pre-IND meeting with the FDA. We've also met with Health Canada. And as you can see here, we're planning to submit our IND application to begin human clinical trials and hopefully, with the agreement of the FDA, begin implanting patients in the first half of next year. The first trial will begin right in patients with type 1 diabetes. It's planned to be a 2-cohort study. The first one, just a few patients, really just looking for safety elements of this. Then we will dose escalate to look for clinical benefit and hopefully even achieve insulin independence in these first patients. So having been working on this project for 11 years at the company, I am just overjoyed to see it come to this final point of proof of concept testing where we can see if we're really taking this ability to cure mice and will it extend into the human patients. And the CIRM has been an amazing supporter over this period of time. Juvenile Diabetes Research Foundation has also been a big supporter. And the financial contributions they've made have been leveraged by patient investors that we've had, one of which I see sitting right here in front. And it really is, drug development is a very expensive endeavor, so we know how important it is to have pools of money like Prop 71 enabled to really take these innovative therapies that right now are so risky, pharmaceutical companies are not really embracing them. We need these other mechanisms to pull them forward. So the patient advocacy, CIRM, patient investors are all part of this equation. And just in summary, as Brooklynn attested to, our dream for this product and the way it's designed is really to meet these unmet needs of type 1 patients. So allowing them hopefully to, if not completely eliminate their insulin syringes, certainly to better manage their blood glucose and hopefully avoid the long-term complications that are really inevitable of the disease eventually. So I won't talk through all of these points here, but I would just like to stop there in the interest of time and thank you for your attention.